In our last lecture, we looked at basic principles underlying sensory function, the mechanisms animals use to obtain information from their environments. In a sense, sensory systems represent input devices that provide data for physiological information processing systems, including the endocrine system and the nervous system. Today we're going to turn our attention to the output side of things. Now that we've looked at sensory receptors, we'll turn to considering the effectors that animals use to respond adaptively to this information. Remember from our discussion of homeostasis that we defined an effector as any mechanism an organism has available to change its physiological state in response to information it receives and processes. For example, when we discussed the endocrine system, I gave you an example of how vertebrate animals regulate their blood calcium levels through the opposing effects of a couple of hormones, calcitonin and parathyroid hormone. In this case, bone cells served as one kind of effector through their ability to change the amount of calcium that they store or release into the bloodstream. The intestine and the kidney were other effectors in this example, which, because of their ability to modify the amount of calcium that's taken in from food or that's excreted, again in response to hormonal signals. Obviously, the ability of an organism to change any number of similar physiological parameters is essential for it to be able to homeostatically regulate its internal environment. But one very, very effective way for an animal to improve its circumstances overall is simply to be able to move. We saw an example of this as well when we talked about thermoregulation in ectothermic animals. You'll remember in that example that we saw how a lizard, which lacks physiological effectors for generating heat internally, can nonetheless maintain its body temperature within a fairly narrow range of values simply by moving in and out of the sun. And as we saw in the example I gave of moths and bats last time, the ability to move is imperative for a whole range of things that an animal has to do to survive and reproduce. To this end, an important and prominent set of effectors that an animal has at its disposal are those that enable it to simply move. These effectors are obviously what we call muscles. What I want to do today is to look briefly at how muscles work. To begin with, we want to sort of consider what it is that muscles do. Now, actually, this is fairly simple because muscles are made of cells that do nothing but change their shape. In fact, the only way they change their shape is in one dimension. They contract. They become shorter. In vertebrate animals, such as you and me, there are actually a variety of different kinds of muscles. For example, we have something we call smooth muscle, which is the kind of muscle that provides contractive forces for our internal organs. We have cardiac muscle, which is a special kind of muscle that, as the name suggests, our hearts are made out of. And we also have what are called striated or skeletal muscles, which are the muscles we use to move, the muscles in our limbs, our trunk, our neck, and so forth. There are many other kinds of muscle cells found in other kinds of animals, but the good news is that the molecular mechanism involved in contraction for all of these kinds of muscle cells is basically the same. Now, what we're going to do for the sake of time is focus our discussion and look at this mechanism in the context of striated muscle in specific. Now, striated muscle gains its name from the fact that it has a very distinctly banded pattern or striated pattern when you look at it under the microscope. And we're going to return to this point and look at the reason for this in a minute. What we need to do to understand how striated muscle functions is first to have a little bit of a look at the anatomy of a striated muscle. Now, a muscle in general can be thought of as a very hierarchically structured organ. And I want to look at the levels of the hierarchy of a muscle. If we dissect a muscle, say if you take a muscle in your arm and you cut through it crosswise, you do a cross section of it, you'd see that the muscle itself is made of a whole bundle of what are called muscle fibers. Now, these muscle fibers are actually cells. Those are the basic cellular unit of muscles, and they're very elongated cells. A single muscle fiber will run from one end or to the other of the entire muscle. These are actually multinucleated cells because developmentally muscle fibers are created from the fusion of a whole series of cells along the length of the muscle. Now, let's take one of these muscle fibers and cut through it crosswise. So now we're cutting through a single cell. If we looked at the cross-section of a single muscle fiber, we'd see that it is in turn made up also of a bundle of units, which we call myofibrils. Now, of course, at this point, you'd actually need a pretty good microscope to be able to see these structures. But in each muscle cell, in each muscle fiber, there are a series of what we call myofibrils, again, running along the length. Now what I want to do is to consider what it would look like if we dissected out one single myofibril, this subcellular structure, and instead of looking at it in cross-section, first let's take a look at it from the side. So imagine we've taken one of these subcellular threads, if you will, out of one muscle fiber, and we're now looking at it under a good microscope from the side. What you would see if you did this was the banded pattern, the striated pattern for which these muscles gain their name. Along the length of the myofibril, you would see a repeating series of light and dark bands, with a very narrow dark line separating each repeating set of bands. So what I want to do now is just give you some names to attach to these structures, because these names will help us sort things out as we go along. The repeating unit, this repeating unit that we see along the length of this subcellular myofibril, is called a sarcomere. The ends of the sarcomere are defined by these very dark, narrow bands, and we're going to call those the Z lines. Now, I didn't make these names up. These are the names that biologists use to describe these, but we want to keep them straight just so that we have something as a point of reference. The sarcomere, which is defined by these Z lines, is the basic functional unit of muscles. So what we want to do is to focus in on how a sarcomere works. Actually, the word sarco is from the Greek root for flesh, and mere is a root for meaning basically part. So sarcomere basically means flesh part. It's the functional unit of how cells work. Okay, now let's get back to looking at the hierarchical organization of muscle function by considering what it would look like if we now cut a cross-section through a single myofibril. Now, these are the subcellular units that are running along the muscle cells, or what we call muscle fibers, okay? Now we're going to cut through a myofibril. Now, you can imagine, given that the myofibril has a banded pattern along its length, that where we cut might determine exactly what we see. But on average, if we cut through a myofibril, we'd see, and we need a really good microscope at this point, we'd see within the myofibril there's again a bundle of elements that we call myofilaments. Now, we've got a hierarchy here. We've got our myofiber, we've got our myofibril within that, and within the myofibril we now have myofilaments. These myofilaments actually are the smallest unit we need to consider. The myofilaments are themselves actually chains of molecules. In fact, these structures are long chains of proteins, little proteins that are all strung together. There are two kinds of myofilaments. There are the so-called thin filaments. And these are chains of a small globular protein called actin. You can imagine each thin filament as looking something like two chains of beads that are sort of twisted together to make a long composite string, with each bead being an actin molecule. The other kind of myofilament is called the thick filament. And these also are made of chains of proteins, but in this case it's a protein called myosin. Now, individual myosin molecules have a more complex structure than actin molecules. This is a point we're going to come back to in a second. But you can still imagine a thick filament as being made up of a chain of myosin molecules that are all woven together. All right, so now what we've done is we've taken the muscle apart all the way down to the molecular level, and we've seen that this functional unit, the sarcomere, has within it a couple of basically strands of chains of proteins. I shouldn't say a couple. There are actually hundreds of thousands of them that are all running lengthwise in that sarcomere. Now let's go back to thinking about what the banding pattern that we saw when we looked at one of these individual sarcomeres from the side comes from. What we see is a pattern of light and dark regions along the length of a single sarcomere. Those light and dark regions correspond to where the thin and thick filaments occur. Specifically, at the ends of the sarcomere, there's a light region. Actually, a light region really on either side of the Z line, but if you just consider one sarcomere on the insides of the Z line that define the end of that sarcomere, there's a light region. This light region is a place where only thin filaments occur. Only the actin thin filaments. It's light because these thin filaments are, as the name suggests, thin. They're smaller molecules. These light regions we're going to give a name to as well, because we'll need that in a second. We're going to call those I bands. Now, in the middle of each sarcomere is a much darker region, a broad darker region that we call the A band. The A band is defined where the myosin thick filaments occur. So we're looking at the side of a sarcomere, there's a dark band in the middle, and this is where the thick filaments are occurring, and there's a light band on either end, and this is where the thin filaments are occurring. It turns out that the thin filaments actually extend into the A band partly as well, and the reason the A band is dark is not only because there are the thick filaments in there, but also thin filaments are extending into that region as well. Now, this banding pattern, then, to review, suggests the following structure. A sarcomere has a plate of protein, structural protein, at either end. This is the Z line. Extending in from that plate of protein are a whole series of these molecular chains, the actin chains, that are called the thin filaments. In the middle of the sarcomere, just sort of sitting there in the middle, are a series of thick filaments that are made of myosin. Now, this is a lot of microanatomy to keep track of. Why are we bothering with all this? The reason is that the overlapping pattern of thin and thick filaments, which we see in terms of these banding patterns under a microscope, actually provided the key to how the sarcomere works on a molecular level. And that's what I want to tell you about now. In 1954, a couple of pairs of researchers, Hugh Huxley and Jean Hansen, who were working in London, and Andrew Huxley and Ralph Niedergerker, who were working in Cambridge, independently proposed a theoretical model that we now call the sliding filament model, which was a model for how muscles contract and was explicitly developed based on looking at microscopic descriptions of how these banding patterns changed during contraction. Now, it turns out, just as an aside, that Hugh Huxley and Andrew Huxley, in spite of their last name, were unrelated. Andrew Huxley, in fact, was a very well-established scientist at the time who had previously been one of the two people to work out how action potentials work on a cellular level. Now, both these sets of researchers noticed the following. They noticed that when a muscle contracts, the distance between the Z lines, that is, the length of the sarcomere, shortens. This makes sense, because if the sarcomere is the functional unit of contraction, then somehow that sarcomere has to become shorter overall. What they also saw was that the length of the I bands also shortens. But remember, the length of the I bands are the light regions on the end of each sarcomere. But the length of the A band, that is the dark band in the middle, didn't change at all. Now, how did they propose to account for these changes in appearance? What they suggested with the sliding filament model was that the thick and thin filaments in the sarcomere actually didn't change their length at all, but slid by each other, and that the change in the degree to which you would see light and dark patterns represented the degree to which these filaments were overlapping. When the muscle was relaxed, you would have regions on either end of the sarcomere where there were only thin filaments, and these would be the light regions that we call the I bands. As the muscle contracted, the thin filaments would slide past the thick filaments, pulling in the Z lines and shortening the sarcomere. As a result, you would see a disappearance of the light region on either end of the sarcomere because there was a decreasing area of no overlap, or I should say an increasing region of complete overlap of the thin and thick filaments. You would see that the A band now basically occupied the entire length of the sarcomere because the regions at the end where the thin filaments had originally been alone disappeared as they slid the two Z bands in towards the center. To the two Z lines, I should say. Now, the implication of the sliding filament model is not just that these thin and thick filaments slide past each other, but more importantly, and actually critically, that somehow there's an interaction between the thin and thick filaments, that is, the chain of actin molecules and the chain of myosin molecules, that not only causes them to move past each other, but does so while it's generating force. Because remember, that's what a muscle has to do. It doesn't just shorten, it shortens in a way that can actually pull on something. That's what we do with muscles. So the movement of the filaments next to each other must somehow involve force generation. How could these two kinds of proteins, the thin and thick filaments, the actins and the myosins, interact in a way that generated this force? Well, the answer comes from looking at the structure of myosin in a little bit more detail. Remember I told you myosin has a more complex shape than the globular actins that make up the thin filaments. Myosin is the molecule that makes up the thick filament. Myosin has what you might imagine is sort of a long tail region, it's called. And this is the part of the molecule that gets woven together with the tails of other myosins to make the thick filament. But myosin also has a head region that sticks out from the side of the thick filament. Now you can think of a thick filament then as a chain of many myosins with all of their heads sticking out along the length of that chain. As we've seen to be the case for proteins in many other contexts, the shape of the myosin molecule can change, and it changes depending on whether or not it's phosphorylated. That is, depending on whether or not it has a phosphate group attached to it. Specifically, when a myosin is phosphorylated, its head region essentially bends forward, and when it's unphosphorylated, the head region bends backward. Now, to picture this, think of your arm as a myosin molecule. And your forearm and hand would be the myosin head region, with your upper arm being the myosin tail. So you might imagine your arm as being one myosin molecule, and if you had a bunch of other upper arms woven together along some chain with other people, then you might imagine that as a thick filament. But your hand is reaching out, and that's the head of the myosin molecule. When the myosin molecule is phosphorylated, or what we would refer to as in its high energy state, your arm reaches out with your palm upward. When it's unphosphorylated in its low energy state, it bends at the elbow and pulls back. It's a cyclic change in the configuration of the myosin head, bending forward and bending backward as it becomes phosphorylated or unphosphorylated, that is the molecular basis for contraction. Now, we've talked about protein phosphorylation earlier in our discussion of how protein kinases work in the context of intracellular signaling. But I want to add one more detail here, and that is that in many cellular processes, phosphate groups are donated by a molecule called ATP, which stands for adenosine triphosphate. We're going to learn a lot more about ATP later in the course when we talk about energy in detail. For now, you just need to know that it's a common currency that's used to store and transfer energy in cellular functions through its ability to donate phosphate groups. Another point is that in many cellular processes, ATP often donates its phosphate group to another molecule in a two-step process. First, ATP binds as a whole to this other molecule, and then once ATP is bound, then a phosphate group cleaves off the ATP, attaches to that molecule, and phosphorylates it. The ATP at that point we would refer to as ADP, adenosine diphosphate, because it's lost one phosphate. Now, the only important thing to keep in mind now is that the ATP needs to bind to the myosin, and then once it's bound, it donates a phosphate group. Myosin can only bind this ATP when it's in its low energy configuration. In other words, when it's bent back, if we're thinking about our arm. This is the configuration that allows it to bind a molecule of ATP. Once ATP has bound, ATP phosphorylates the myosin head and it bends forward. Now, the interesting thing here is that when myosin has its head in the forward position, in its high energy position, it can also bind to a special site on actin molecules. So imagine your arm reaching out, and once it's reaching out, it can reach up and grab an actin molecule or a binding site on an actin molecule on a thin filament nearby. The binding of the actin molecule to this myosin itself triggers a configurational change in the myosin head, causing it to lose its phosphorylation. It loses its phosphate and the ADP. They drop off. Well, when it's no longer phosphorylated, the myosin head returns to its low energy configuration. It bends back. But it's still got hold of the actin thin filament. And so, because it's reached out, bound to an actin on a thin filament, lost its phosphate, returns to its other configuration, it literally pulls that actin along and actually ratchets that thin filament just a few nanometers, like 5 or 10 nanometers, but enough to actually move the filaments past each other under force. Now, this is actually a cyclic process because what happens is if we're thinking again of just one myosin attached to just one actin, we've now got a myosin head that's reached out in its high energy form, grabbed an actin, loses its phosphate, bends back in its low energy form. Now that it's in its low energy form, it can bind another molecule of ATP. Remember, that's the configuration that binds ATP. It's still got a connection to the actin. We call those connections, by the way, cross bridges. But when it binds ATP, this causes another configurational change in the myosin molecule, and it causes the cross bridge to break. So it's brought an actin along with it, but now that it's in its low energy configuration, it can bind ATP again, and that causes that cross bridge to break. Well, now that it's got ATP bound, it's no longer connected to an actin, the ATP phosphorylates the myosin head, it reaches out, it can grab another myosin, it loses its phosphorylation, it pulls back, it's pulling the myosin along with it. Grabs another actin on the thin filament, it pulls the actin along with it, it then binds another molecule of ATP, it drops off, and so on and so forth, with this myosin head essentially reaching out, grabbing a binding site on a thin filament actin molecule and pulling it back repeatedly in a cyclic fashion. And this is the molecular basis of how these thin and thick filaments slide past each other. There's an interesting consequence of this mechanism of contraction on the molecular level. Remember, I told you that the myosin head, when it's bent back in its low energy configuration, can bind ATP, and when it does, it'll let go of the thin filament actin that it happens to be attached to. If there's no ATP present, then it won't let go. It holds on. Now, an interesting concept, usually there's plenty of ATP in muscle cells. So usually it will always find another ATP and it will let go and the process repeats. But if an organism dies, it runs out of ATP. And what happens is that myosin heads reach up, grab actins, return to their low energy configurations, and they can't let go. The muscle becomes stiff. This is the condition that on the whole body level we refer to as rigor mortis. You've basically run out of ATP, and that makes these cross bridges not able to break. Well, this molecular interaction is really remarkable because it's an exquisite example of how shape changes of proteins create a very, very important function. This myosin head is changing its shape in response to a number of different binding events. First, the phosphorylation, then binding an actin site, then losing the phosphorylation, returning to its low energy configuration, binding an ATP, letting go of the cross bridge, and so forth. It's a series of changes of shape that are responsible for this movement of filaments, which is the fundamental way that animals have available to create movement. There's a question, though, left unanswered here, and I want to turn to that briefly now. And that is, if this interaction between myosin and actin is responsible for muscle contraction, what turns the mechanism on and off? I mean, we know that our muscles aren't always contracting. Sometimes they're relaxed. So we should be able to contract or not contract muscles. What is it that controls the interaction between myosin and actin and in that way turns on or off muscle contraction? Well, to understand this, we need to spend just a little bit of time talking about the electrical properties of muscle cells. And this won't take much time because basically, to a first approximation, muscle cells work a lot like neurons work. They have voltage-gated channels, sodium and potassium channels, just as neurons do, and as a consequence of that, muscle cells can fire something that's not too different from an action potential. Now, we know just from anatomy that muscle cells will fire when the neurons attached to them are sending them signals. The attachment between a neuron and a muscle is something we call a neuromuscular junction, and it's actually a kind of chemical synapse. So a neuron can talk to a muscle cell and tell it to turn on and off by firing that neuromuscular junction, and in so doing causing a depolarization in the muscle cell that's similar to the kind of depolarization we might see in an action potential. It's actually different from an action potential in many ways, but it's nonetheless a very rapid and extreme depolarization that then returns back down to a resting level after just a few milliseconds. The question then becomes, how is it that this depolarization that we know happens in muscle cells controls the interaction between actin and myosin? And the answer to this question has to do with yet another set of proteins. These are proteins called tropomyosin, and actually a complex of proteins that we can simply lump together and refer to as the troponin complex. Now, let's go back to picturing an actin thin filament. Remember I told you it's like two strings of beads that are sort of loosely twined around each other, right? And each bead we now know has a binding site that a myosin molecule can reach, a myosin head can reach up and grab and attach to. Well, it turns out that the tropomyosin molecule, this other protein, is also wound around the actin thin filament. And it's wound around the actin thin filament in such a way that the tropomyosin is blocking the binding sites that myosin would ordinarily bind to. So picture tropomyosin as basically being a thread that's winding around along with our beads, and if there was a binding site in a particular position with every bead oriented along that spiral, the tropomyosin would be occurring right over that binding site. So with tropomyosin in association with an actin thin filament, myosin simply can't bind. Why? Not because myosin can't move, it can move just fine. It's just that it can't reach up and grab a binding site. The binding sites are covered. Well, you can imagine then the way that we can control muscle contraction is by somehow controlling whether those binding sites on actins are covered or uncovered. How does that work? Well, this is where that other protein or set of proteins, the troponin complex, comes into play. Now, the troponin complex more or less sits on the tropomyosin. So now we're going to add one more element to our string of beads that's the actin thin filament. Imagine now that you have troponin complexes as sort of beads that are arrayed sparsely along sort of on the troponin, little, like little balls hanging off the troponin, the tropomyosin as it winds around the actin thin filament. The troponin complex is able to bind calcium. Once again, we see calcium playing an important role here. If the troponin complex binds a calcium ion, that binding of calcium causes the troponin complex to change its shape, as we've seen many times before. The troponin complex changing its shape causes the tropomyosin to change its shape, which reveals the binding sites on actin. So the way that these binding sites on the actin become revealed is through the presence of calcium causing configurational changes in the troponin complex, which causes configurational changes in the tropomyosin. Well, where does the calcium come from? Well, here's where yet another set of voltage-dependent channels becomes important, because it turns out that muscle cells have voltage-dependent or voltage-gated calcium channels that are arrayed in occur in very high density on the surface of a cellular structure called the sarcoplasmic reticulum. Now, we learned a while ago about the endoplasmic reticulum, you'll recall. Sarcoplasmic reticulum has a similar name. It's a very different structure, unique to muscle cells, and it is essentially a storage site for calcium. There are pumps, active ion pumps, that pump calcium into the sarcoplasmic reticulum. So the sarcoplasmic reticulum has a very high concentration of calcium, and in a typical muscle cell, there's virtually no calcium out in the cell itself. When the muscle cell depolarizes as a result of an action potential firing at the neuromuscular junction, this causes these voltage-dependent calcium channels to open, and calcium rushes out of the sarcoplasmic reticulum very quickly. It binds with the troponin complex, causing the actin binding sites to be released, and therefore the muscle contracts. Almost as quickly, when depolarization ends for the muscle cell, the calcium is very quickly sequestered back into the sarcoplasmic reticulum. Again, the binding sites are closed and the muscle is no longer contracting. Another example of the role of calcium, of the role of shape changes, of the interactions, the complex interactions among proteins that allow these cellular functions that, in fact, we take for granted. As I'm moving up here, as you're moving out there, it's all about calcium, voltage-gated channels, the movements of proteins. Well, Like sensory perception that we discussed last time, the control of muscular movement depends on many aspects of an animal's anatomy and physiology that we really haven't had time to talk about. But there is one last point I want to end with, and that is that in most cases, contraction of muscle itself can't account for animal movement. All the muscle does is shorten. What muscles really do is shorten the distance between hard parts that they're connected to. Hard parts we call skeletons. So to really understand animal movement, we can't just look at muscles. We have to put those muscles, the muscle cells, the molecular mechanisms, into the context of the whole organism and how it's connected with other parts of the organism, providing us with another reminder of how we can't fully understand any one system in a complex organism unless we consider how that system fits together with the remainder of the organism.